Welcome to video 3 of the Chemistry for Biologists series, Lipids. In this video, we'll explore the structure of lipids, the different types found in organisms, and their vital functions in the body. If you're finding these lessons helpful, don't forget to subscribe to my channel Bio by Tash for more videos in this series. Let's dive in. Lipids are a diverse group of hydrophobic molecules. They are large biological molecules, but not considered polymers or macromolecules. Lipids are made up of carbon, hydrogen, and oxygen. The ratio of hydrogen to oxygen is not 2 to 1, unlike in carbohydrates. Instead, hydrogen is present in larger amounts. Lipids contain much less oxygen than carbohydrates. They are insoluble in water, hydrophobic, but soluble in organic solvents like alcohol and ether. Due to their structure, lipids are efficient energy stores and play structural and functional roles in cells. Fats and oils are two important types of lipids. Chemically, they are very similar, but differ in their physical state at room temperature. Fats, example, butter, are solids. Oils, example, olive oil, are liquids. Sources. Fats mainly come from animal sources. Oils are primarily obtained from plant sources. Fats and oils are made of two main types of organic substances, fatty acids and glycerol. These two are combined using ester bonds, formed in a condensation reaction, removal of water. Glycerol is an alcohol that contains 3 carbon atoms, and each carbon is attached to a single hydroxyl, OH, group. Fatty acids are hydrocarbon chains typically consisting of 16 to 18 carbon atoms, with a carboxyl group at one end of the chain. Living tissues contain more than 70 different types of fatty acids. How variations occur in fatty acids. 1. Length of the carbon chain. Most fatty acids have chains that are typically 15 to 17 carbon atoms long. 2. Degree of saturation, number of double bonds between carbon atoms. There are two groups of fatty acids depending on the degree of saturation. Saturated fatty acids. Each carbon atom is joined to the next by a single covalent bond. These fatty acids have no double bonds. Example, stearic acid. 2. Unsaturated fatty acids contain one or more double covalent bonds between carbon atoms. These are two types. 1. Monounsaturated fatty acid has one carbon-carbon double bond. 2. Polyunsaturated fatty acid has more than one carbon-carbon double bond. Example, linoleic acid. A polyunsaturated fatty acid is an essential fatty acid. It cannot be synthesized by the human body. It must be obtained through the diet. Ester bond. An ester bond is a type of covalent bond formed between a carboxylic acid group and an alcohol group during a condensation reaction. A reaction in which a molecule of water is removed. This is known as esterification. In biological systems, ester bonds are most commonly found in lipids, especially in triglycerides. Triglycerides. Triglycerides are a type of lipid used mainly for long-term energy storage in both animals and plants. They are the most common form of fat found in the body and in foods. A triglyceride is made up of 1 glycerol molecule and 3 fatty acid molecules. These components are joined together by ester bonds, formed through a condensation reaction. Phospholipids. Phospholipids are a special type of lipid that are essential components of cell membranes. They are structurally similar to triglycerides, but with a key difference. That is, one of the 3 fatty acids is replaced by a phosphate group. A phospholipid molecule consists of 1 glycerol molecule, 2 fatty acid chains and 1 phosphate group, which is often bonded to another small polar molecule, like choline. Glycerol with phosphate group makes the hydrophilic head part of the phospholipid molecule, while 2 hydrocarbon chains make the hydrophilic tail part. The phosphate head carries a negative charge, soluble in water and hydrophilic, which means water-loving. The fatty acid tails are neutral, insoluble in water and hydrophobic, which means water-hating. The amphiteric nature of phospholipid causes them to arrange themselves in specific ways when placed in an aqueous, water-based environment. Two common structures are 1. Monolayer. A monolayer forms when phospholipids are spread on the surface of water, such as in experimental setups or simple lipid-water interfaces. In this arrangement, the hydrophilic, polar, phosphate heads face downward into the water, attracted to it. The hydrophobic, nonpolar, fatty acid tails point upward into the air, away from the water. This forms a single layer of phospholipid molecules. It demonstrates the amphipathic nature of phospholipids and how they orient themselves to minimize energy in watery environments. 2. Micelles, sometimes called mezels. Micelles are spherical clusters of phospholipid molecules that form when phospholipids are completely surrounded by water. In a micelle, the hydrophilic phosphate heads are oriented outwards, facing the surrounding water and interacting with it. The hydrophobic fatty acid tails are folded inward, away from the water, hidden in the center of the sphere. This structure is energetically stable in water because water interacts only with the polar heads. The nonpolar tails are shielded from water. Biological roles of lipids. Energy storage. Triglycerides store about 3 times more energy per gram than the same mass of carbohydrates. This is due to the high number of CH bonds, which release large amounts of energy when oxidized. Energy source. During aerobic respiration, lipids are oxidized, breaking CH bonds to release energy. Products of lipid oxidation: carbon dioxide, water, ATP, a high-energy molecule used for cellular activities. Component of cell membranes. Phospholipids form the bilayer structure of all cell membranes. They contribute to membrane fluidity and permeability. Insulation and protection. Thermal insulation in mammals, example, under the skin, shock absorption for internal organs. Waterproofing. Waxy lipids help in water retention in plants and insects. Hormone production. Steroid hormones, example, testosterone, estrogen, or lipid-based molecules. Thank you for watching. If you found this video useful, don't forget to subscribe to my channel. The next video will be all about proteins, so stay tuned.